When you look at a blueprint of a building, it's incredibly precise.
Right. Yeah. You see every single detail.
Exactly. You see the steel girders, the concrete foundation, the exact load-bearing walls. And if you understand that blueprint, you know exactly how the building stays standing.
You can see the whole structure.
Yeah. But when you hand a medical student or, say, a young professional, or really any curious learner, a prescription pad, the blueprint of what they're actually doing is completely invisible.
Oh, absolutely. It's totally hidden.
Right. You are prescribing this microscopic chemical to interact with a highly specific receptor inside a cellular membrane in a body made of trillions of cells. It is basically the ultimate invisible architecture.
It really is. And because we're so used to seeing the macroscopic clinical outcome, the blood pressure drops, the fever breaks, the pain fades.
The stuff we can actually measure.
Right, the visible stuff. Because of that, it is dangerously easy to forget the microscopic physico-chemical warfare and, negotiation happening to achieve that result.
Yeah, warfare is a good word for it, because every time you write a prescription, you are launching a microscopic chemical war. So today, we are looking at how to design the perfect molecular weapons.
I love that analogy.
Welcome to this deep dive. Whether you're prepping for ward rounds or, studying for your boards or just insanely curious about how medicine actually works, we are taking the dense fundamental concepts of medicinal chemistry and translating them into a masterclass.
A very practical masterclass, hopefully.
Definitely. We're going to look under the hood at how drugs are discovered, how they physically interact with our biology, and most importantly, how you apply that directly to clinical practice.
And I think a really strong place to anchor this right at the start is with the official definition.
Lay it on us.
So the International Union of Pure and Applied Chemistry, IUPAC, they define medicinal chemistry not just as making chemicals.
Right. It's not just mixing things in a beaker.
Exactly. They define it as the discovery, development, identification, and interpretation of the mode of action of biologically active compounds at the molecular level.
Which is a mouthful, but it's basically about understanding the underlying why and how, right?
Yeah, exactly. And getting to a final, safe clinical drug involves three really foundational stages. First, you have the discovery or identification stage.
Finding the lead molecule.
Right. You find a new active substance. Then comes optimization, where you take that lead and you hack its structure to make it more potent, more selective, and hopefully less toxic.
And then the final stage.
The development stage. That's where scientists figure out how to mass produce it and refine its pharmacokinetics so it behaves, predictably in the human body.
Okay, let's unpack this. Because before a drug ever reaches that high-tech optimization stage in a pristine lab, it has to originate somewhere. Right?
Yeah, it doesn't just appear out of thin air. Early medicinal chemistry was primarily concerned with isolating active agents from plants. But nature's pharmacy is, it's far more diverse than just flora.
I always find it fascinating that some of our most powerful chemotherapeutic agents against infective diseases came directly from the dirt.
Literally from the soil, yeah.
Right. Soil fungi and bacteria gave us penicillin, streptomycin, and tetracycline.
Which changed the world.
Completely.
Yeah.
And if you think about the evolutionary biology behind it, those microorganisms, they weren't making antibiotics for human benefit. They were fighting microscopic turf wars in the soil, engineering chemical weapons to kill competing bacteria. We just isolated their weapons.
We absolutely did. And we also pulled medicinal compounds directly from minerals.
Oh, like inorganic stuff.
Yeah. Elements like iron, calcium, and magnesium have obvious biological roles, but historical records show humans leveraging mineral-containing medicated hot springs for centuries.
Oh, wow, really?
Yeah, specific geological locations in India, like the springs at Rajgrin in Bihar and Shrahara in Dehradun, were early crude forms of delivering mineral-based therapeutic agents.
That is wild. So we source these raw chemical weapons from fungi, plants, or, literal mineral springs. But once you put them into a human body, what are they actually doing to a cell?
Well, there seem to be two main mechanisms of drug action. The first is enzyme inhibition.
Okay, so stopping a reaction.
Right. You introduce a drug that modifies normal biochemical reactions. It might bind reversibly or non-reversibly to an enzyme, or you might use an antimetabolite.
Well, which is like a decoy, right? A molecule that mimics a natural substance to trick the enzyme into binding with it instead.
Exactly. It jams the machinery. The second major mechanism is the drug-receptor interaction, where drugs act on the cell membrane through physical or chemical interactions at specific receptor sites.
Now, I have to stop you there because whenever I hear receptor site, my mind immediately defaults to the classic lock and key analogy from high school biology.
Oh, sure, everyone learns that one.
Right. The receptor is a rigid lock on the cell membrane, and the drug is a perfectly shaped physical key that snaps into it. But I find that metaphor a bit misleading. Is it really just a perfect physical geometric fit, or is there a deeper chemical drive forcing that key into the lock?
That's a great question. And what's fascinating here is that the lock and key metaphor kind of falls apart when you look at the thermodynamic reality of the cell.
How so?
What really drives that binding is heavily dependent on hydrophobic forces. Most drug molecules have a non-polar lipophilic portion.
So fat-loving groups.
Yes, exactly. Fat-loving groups like alkyls or aryls. And remember, the cell membrane itself is a lipid bilayer.
Right, and water and oil don't mix. The human body is mostly water, so these fat-loving drug molecules are essentially trying to escape the water.
Exactly the opposite of a mechanical lock. The chemical affinity, the thermodynamic desire of these nonpolar drug regions to associate with the nonpolar regions of a receptor simply to get away from the aqueous blood environment is the crucial driving force.
Wow, so they're just fleeing the water.
Yeah. The physical shape matters for selectivity, obviously, but it is this chemical aversion to water that pushes the drug into the receptor in the first place.
That makes so much more sense. It's chemical attraction and repulsion, not just a puzzle piece sliding into place.
Yeah.
But even if a drug has the perfect shape and the perfect hydrophobic drive, its physico-chemical properties dictate whether it can survive the journey through the hostile environment of the human body.
Oh, absolutely. Solubility, partition coefficients, dissociation constants, they all dictate a drug's survival.
It's a gauntlet.
It really is. But let's examine a very specific life-saving property, chelation. Chelation occurs when a drug molecule donates electrons to a metal ion, forming a ring structure.
So it basically traps the metal.
Exactly. The drug acts as a ligand, essentially building a chemical cage around the metal.
Which is exactly how we treat heavy metal poisoning. Like, dimercaprol is a chelating agent used as an antidote for organic arsenical poisoning, specifically from compounds like lewisite.
Yes, and it's highly effective.
Right, and it also traps antimony, gold, and mercury. You inject the dimercaprol, it hunts down the toxic heavy metal, wraps around it to form a water-soluble complex, and then the kidneys just flush it out.
And penicillamine does the exact same thing for copper poisoning.
Oh, right.
And then you have things like 8-hydroxyquinoline, which uses chelation to complex with iron or copper on the surface of microbes.
Wait, on the microbes themselves?
Yeah, which physically disrupts their cellular processes, making it a highly effective antibacterial and antifungal agent.
That's incredibly clever. Then there is surface activity, which relies on surfactants. These are molecules with a split personality. You have a water-soluble hydrophilic head and a water-insoluble hydrophobic tail, usually an 8 to 18 carbon chain.
Right, the amphiphilic molecule.
Exactly. When you put them in a watery environment, those hydrophobic tails squeeze away from the water and cluster together, forming a sphere called a micelle.
And that physical behavior is a potent pharmacological tool. Quaternary ammonium compounds, like benzalkonium chloride, use this surface activity to wedge themselves into the lipid membranes of bacteria.
So they just jam themselves in there?
Yeah, they disrupt the membrane's surface tension, effectively popping the bacteria like a balloon.
Brutal, but effective.
Hexyl resorcinols use the same micelle-forming surface activity for anthelmintic effects, destroying the outer layers of parasitic worms.
Okay, but the physico-chemical interaction that completely changes how you manage a patient on the wards is protein binding. When a drug enters the blood plasma, it often binds to circulating proteins, right?
Yes, very frequently.
Acidic and basic drugs tend to bind to serum albumin via dispersion forces and hydrogen bonding, while some basic drugs bind to acid glycoprotein.
And this is a highly calculated part of drug design.
I am going to challenge that, actually. Because logically, if a drug is physically stuck to a massive plasma protein, like albumin, it's trapped in the bloodstream. It cannot cross the phospholipid bilayers to enter tissues. It cannot pass through the glomerular membranes in the kidneys to be excreted, and it certainly cannot interact with its target receptors. Aren't you essentially neutralizing your own drug before it can even work?
Well, you are neutralizing a portion of it, yes, but that is a deliberate, vital, protective mechanism. We call it the depot effect.
The depot effect.
Right. Because the bound drug is trapped in the bloodstream, it is shielded from the liver enzymes responsible for first-pass metabolism, and it is protected from being rapidly filtered out by the kidneys.
I see. So the protein acts as a biological storage tank.
Exactly. As the free, unbound drug does its job and is eventually cleared from the body, the drug-protein complex, which is reversible, by the way, releases more of the drug to replenish the free concentration in the blood.
So it creates a sort of slow release.
Yes. This gives the drug a prolonged, steady half-life rather than a massive, dangerous spike followed by an immediate crash.
But that balance creates a terrifying clinical trap. If a patient takes a standard dose of a highly protein-bound drug, say 90% of it binds to albumin and 10% is free to act, if that patient develops severe hepatic disease, their failing liver stops producing albumin.
Or if they develop uremia from kidney failure, which fundamentally alters the binding affinity of the albumin they do have.
Right. Suddenly you don't have enough functional storage tanks. Instead of 90% of the drug safely bound, maybe only 50% binds. You haven't changed the dose, but the active free concentration of the drug in their blood has just quintupled.
It's a massive spike.
A standard therapeutic dose instantly becomes a lethal toxic overdose.
Which perfectly transitions us into why medicinal chemists spend so much time in that second stage of discovery we mentioned earlier, optimization.
Because nature isn't perfect.
Right. When nature's original blueprint has flaws like causing severe toxicity or being chewed up by metabolism, too quickly chemists have to hack the molecular structure. And one of the most elegant methods for doing that is biosaustorism.
Oh, I love this concept.
Mhm.
Irving Langmuir introduced this back in 1919. He realized that if you have molecules or ions with the same number and arrangement of electrons isosters, they should possess similar physical and chemical properties.
Which is a genius observation.
Truly. So if a specific functional group on a drug is causing a side effect, you don't throw the whole drug away. You swap that group out for a biosauster, a piece that looks and acts the same electronically but completely removes the flaw.
Yeah, we use classical monovalent replacements quite a bit, like swapping a hydrogen atom for a fluorine atom.
Oh, like in cancer drugs.
Exactly. Consider the anti-neoplastic agent 5-fluorouracil. Fluorine is roughly the same size as hydrogen, so the body's enzymes still recognize the molecule. But fluorine is highly electronegative.
So it acts like a trap.
Right. When the enzyme tries to process the 5-fluorouracil, the fluorine completely jams the enzymatic machinery, shutting down DNA synthesis in cancer cells.
Incredible. And you could also use divalent replacements involving double bonds, right? Like torostat. It's an aldose reductase inhibitor used to treat diabetic neuropathy. Chemists took its structure and swapped a carbon double bond sulfur group for a carbon double bond oxygen group.
Oxytolrostat.
Yeah. The new compound oxytolrostat kept all of its biological activity, but had a completely different, much safer metabolic profile.
But the real ingenuity shines with non-classical biosaustorism.
Here's where it gets really interesting.
It does. Because these are replacements that do not look structurally similar in the traditional steric sense, but they maintain similar electronic properties like their dissociation constant or pK. This is how we outsmart the human body's own defense mechanisms.
The story of isoproterenol is the perfect example of this. Isoproterenol is a prototype beta-adrenergic agonist. It relaxes the smooth muscle in the lungs, making it a great bronchodilator for asthma.
But it has a major flaw.
Right, there is a massive physiological hurdle. The molecule contains a catechol ring with a specific 3-hydroxyl group, and the human body has an enzyme called COMT catechol O-methyltransferase, whose singular purpose is to find that exact structure and destroy it.
So if you give an asthma patient isoproterenol, they can breathe easily for a few minutes, but because COMT shreds the drug almost instantly, they are wheezing again 15 minutes later.
It's useless as a long-term treatment.
Yeah.
So chemists looked at that 3-hydroxyl group and realized they needed a molecular disguise. They needed to swap it for something the lung receptor still recognized as a key, but that the COMT enzyme was completely blind to.
And they used non-classical biosaustorism to do it.
Exactly. By swapping the hydroxyl group for a hydroxymethyl group.
And that simple change created albuterol.
Which is everywhere now.
Yeah. In another iteration, they swapped it for a sulfonamide group to create sudornol. The lung's beta receptors still bind to these molecules perfectly, providing that critical bronchodilation, but the COMT enzyme completely ignores them. You bypass rapid metabolism entirely.
It is brilliant. Another way chemists manipulate molecules to bypass the body's defenses is the prodrug concept, first proposed by Adrian Albert in 1958.
Yes. A prodrug is a pharmacologically inactive derivative of a drug.
I like to think of a prodrug as a piece of flat-pack furniture.
Okay, I like where this is going.
Think about it. If you buy a massive, fully assembled bookshelf, you might not be able to fit it through the front door of your house. But if you pack it down into a flat cardboard box, it slides right through.
That makes sense.
A prodrug is the flat-pack version. You attach a temporary chemical group to hide the bulky active drug, altering its physico-chemical properties so it can absorb through the gut wall, the front door. Once inside the bloodstream, metabolic enzymes act as the screwdriver, breaking off the temporary addition and assembling the active drug.
That is a highly accurate way to visualize it, actually. You are temporarily altering the molecule to overcome an absorption hurdle or to reduce its toxicity on the way to its target. But this is completely distinct from the concept of soft drugs.
Oh, right. People get those confused.
Yeah, they do. A soft drug is therapeutically active from the very moment you ingest it.
Okay, I have to push back on the concept of soft drugs, though. The premise is that they are designed to have a predictable and controllable metabolism in vivo, breaking down into non-toxic pieces once their job is done.
Right, that's the goal.
But how do you program a reliable self-destruct sequence into a chemical when every single patient's liver function and metabolic rate is wildly different?
Well, the trick is you don't rely on the liver.
Oh.
Yeah. The self-destruct sequence in a soft drug is usually an ester bond carefully engineered into the molecule. The human bloodstream is packed with ubiquitous esterase enzymes that constantly cleave these specific bonds at a highly predictable rate, regardless of liver health.
That was clever.
Very. The drug does its job locally, and the moment it hits the general circulation, esterases hydrolyze it into inactive, harmless metabolites. It prevents systemic toxicity almost entirely.
So we have covered the mechanisms, the physico-chemical properties, and the structural hacks. To really solidify this, let's stop looking at isolated molecules and see how we engineer solutions to specific anatomical hurdles in clinical practice. Let's do a quick systems tour.
I think that's a great idea. Let's take the autonomic nervous system and the physical hurdle of the human eye. To treat glaucoma, you need to reduce intraocular pressure, right?
Right, it's a fluid buildup problem.
Exactly. So we use cholinergic drugs like carbachol. By synthesizing a molecule from ethylene chlorohydrin and phosgene, chemists created a compound that forces the ciliary muscle to contract.
Which induces meiosis, constricting the pupil.
Yes, and that physically pulls open the trabecular meshwork in the eye, allowing aqueous fluid to drain and relieving the pressure.
But sometimes you need the exact opposite effect.
Right.
Like if you were doing an eye exam, you need the pupil dilated. So we use anticholinergics like tropicamide. It acts as an M4 receptor antagonist.
Right, it blocks the signal.
Exactly. By blocking the acetylcholine receptors, it paralyzes the sphincter muscle of the iris. And tropicamide is engineered specifically for this because it has a fast onset and a short duration, meaning the patient isn't walking around blind to bright light for 3 days.
Moving to the systemic anticholinergics, we engineer drugs to overcome gastrointestinal hurdles. Oxyphencyclamine is a peripheral antisecretory agent. It blocks the receptors that cause smooth muscle spasms in the gut.
And clothianidine bromide works similarly, right?
Yeah, it decreases stomach acid production to treat peptic ulcers and calms the hypermotility of a spastic colon.
Let's shift gears to the cardiovascular and respiratory systems. We already explored how albuterol targets beta-2 receptors in the lungs for asthma. Isoatherin is another adrenergic drug synthesized to do the exact same thing, but if we tweak the structure to target alpha receptors instead, we get pressor agents for cardiovascular crises.
Metaraminol and methentramine are prime examples of that.
Lifesavers in the ER.
Absolutely. If a patient is hemorrhaging or their blood pressure bottoms out during spinal anesthesia, you administer these parenteral vasopressors. They force the smooth muscle in the blood vessels to constrict, shrinking the pipes to artificially maintain the blood pressure and keep the brain perfused.
Pain management is another massive hurdle, and it brings us back to endogenous receptors. The opiates are divided into 2 chemical groups. The phenanthrene group, which includes morphine, codeine, and the bane.
And then the benzallocequinolone group, which includes papaverine.
Exactly. And these exogenous opiates are structural hacks of our own biology. The body has endogenous peptide receptors designed for our natural painkillers, endorphins, and enkephalins.
Right, our internal pain management system.
Morphine is simply a molecule engineered by nature to fit so perfectly into those mu-opioid receptors that it intercepts the ascending pain pathways in the spinal cord. But because those same receptors exist in the brainstem and gut, Binding them also depresses respiration and stops gastrointestinal motility.
Which is why opioids have such dangerous side effects. For a totally different approach to pain, we use nonsteroidal anti-inflammatory drugs, or NSAIDs. Antipyrine is a pyrazolin derivative and actually one of the very first synthetic compounds used in medicine.
Oh, really? I didn't know it was that old.
Yeah, and instead of blocking the pain signal in the brain, like opioids, it acts peripherally. When tissue is damaged, enzymes create prostaglandins that cause inflammation and sensitization. NSAIDs inhibit those enzymes, basically stopping the chemical fire alarm from ringing in the first place.
The kidneys present perhaps the most complex physiological engineering challenge. Diuretics are designed to target incredibly specific physical pumps along the nephron. Like in the proximal tubule, you have carbonic anhydrase inhibitors like acetazolamide, which stop the reabsorption of bicarbonate.
You can also use brute force physics?
Like mannitol.
Exactly. Osmotic diuretics like mannitol work primarily in the loop of Henle and proximal tubule. Mannitol is filtered into the urine, but it can't be reabsorbed. Because it is highly concentrated, water is osmotically dragged into the tubule and trapped there, forcing the body to excrete it.
But the heavy hitters are the high ceiling, or loop diuretics. These drugs physically jam the sodium-potassium 2-chloride symport in the thick ascending limb of the loop of Henle.
And that causes a massive shift.
Oh, yeah. If you block salt from being reabsorbed into the body, the salt stays in the urine. Water always follows salt. So the water stays in the urine to be excreted, producing massive peak diuresis. We divide these chemically into sulfonamide derivatives like furosemide and bumetanide, and non-sulfonamide loop diuretics like ethacrynic acid.
Finally, we face the endocrine hurdle of diabetes. Managing blood sugar isn't just about replacing insulin. It's about changing how the body processes sugar, fundamentally.
Right.
Metformin is a masterpiece of multi-targeted engineering. It reduces hepatic glucose output by inhibiting gluconeogenesis, literally stopping the liver from manufacturing new sugar.
Which is huge.
And simultaneously, it forces peripheral muscle cells to become more sensitive to the insulin that is already there, while reducing the absorption of glucose in the intestines.
It attacks the problem from three sides. Then you have the phospholipidones, or glitazones. These are PPAR gamma agonists. They go straight into the nucleus of fat and muscle cells and turn on specific genes that make the cells highly responsive to insulin. But this class of drugs provides a stark historical warning about the optimization phase we discussed at the very beginning.
Yes, the triglytazone disaster.
Exactly. Troglytazone was the first drug in this class, developed and approved in 1997. But the chemists missed a critical flaw in its metabolic pathway. By 1999, it had to be completely pulled from the market because that specific molecular structure caused catastrophic hepatic toxicity.
It just destroyed the liver.
Right. It proves that no matter how elegant the receptor mechanism is, if the chemistry breaks down into a liver poison, the drug is a failure.
It's a sobering reminder of how high the stakes are in medicinal chemistry.
So what does this all mean? We have traveled from isolating active compounds out of dirt and mineral springs, all the way to engineering biososters that act as molecular disguises and prodrugs that unpack themselves inside the cell. Every drug you prescribe, every side effect you memorize is rooted in these fundamental physico-chemical properties.
And to test your grasp on that unseen architecture, I highly recommend you think through a clinical scenario before your next shift.
Oh, a pop quiz. Let's hear it.
If you are managing a patient on a highly protein-bound drug who suddenly develops severe kidney failure causing uremia, how will that uremic state alter the binding affinity of their plasma albumin? And more importantly, how will you adjust your dosing to prevent the sudden spike of free drug from causing toxicity?
That is exactly the kind of critical thinking that separates a good clinician from a great one. Tracing the logic from the molecule to the plasma protein to the patient's failing kidney is how you actually master this material. But I want to leave you with a completely different thought to ponder. We spent this entire deep dive talking about how human chemists painstakingly hack molecules to bypass enzymes and survive the bloodstream. But we're currently standing at the edge of a totally new era.
The AI revolution.
Exactly. With the rise of artificial intelligence and deep learning models predicting protein folding and receptor shapes in milliseconds, how long will it be until the entire optimization phase of medicinal chemistry is obsolete? What happens when an AI can design the perfect, non-toxic bioisoster from scratch, bypassing nature's blueprint entirely? Keep an eye on that horizon. Thank you for joining us on this deep dive.